Hello everyone, myself Dr. Amanpreet Kaur Kalsi. In this lecture, we will take up the topic of proteins. The parts we'll cover in this lecture include introduction to proteins, amino acids, classification of amino acids, peptide bond formation, orders of protein structure, classification of proteins, and biological importance of proteins. The term protein is derived from a Greek word which means primary or holding first place. The proteins, which are nothing but chains of several hundred or more amino acids. These chains of amino acids give rise to approximately 1 lakh different types of proteins, which are all made up of nothing but just the 20 amino acids. These proteins constitute 20% of a human body, and if we leave aside the 60% water content, out of the remaining 40%, 50% of dry weight of human body is made up of proteins. These proteins make almost every part of our body, including skin, enzymes, bones, nails, hair, internal organs, and hormones. The elementary composition of proteins include 50 to 55% carbon, 6 to 8% hydrogen, 20 to 23% oxygen, 15 to 18% nitrogen, and 0.4% sulfur. Now talking about amino acids, which are the building blocks of proteins. There are about 300 amino acids in nature, but only 20 of them take part in protein synthesis. These amino acids in proteins or the peptide chains are linked together by peptide bond, and only L amino acids occur in proteins, where L refers to the configuration of the proteins. This figure shows a basic diagram of protein having an amino group, a carboxyl group, and a side chain R. The amino acids are bifunctional compounds because they have a carboxylic acid group and a basic amino group attached to the same carbon atom. Next, coming to classification of amino acids. According to the polarity of side chain R, amino acids can be classified into 6 groups. First, having aliphatic side chain. Second, with SH group, that is sulfur group. Third, having acidic group or their amides. Fourth, having basic group. Fifth, having aromatic ring in the side chain. And sixth group, having AMINO group at the R chain. First, amino acids with aliphatic side chains. These include glycine, alanine, valine, leucine, and isoleucine. We can see the symbol of each one of them in second column and structural formula for each in the third column. The gray part shows the basic amino structure with the amino group, carboxylic group, and the central carbon, and the unshaded part in the structural formula is the R chain, which in glycine has only hydrogen atom, in alanine has a methyl group, and subsequently in valine, leucine, and isoleucine they have different aliphatic side chains. Valine, leucine, and isoleucine have different aliphatic side chains. Second group with side chains containing hydroxylic group. This include serine, threonine, and tyrosine. The second column shows their symbols and third column shows their formula. Next group is with side chains containing sulfur atoms. These contain cysteine and methionine. Both have sulfur atoms in their side chain. Next group include amino acids with side chains containing acidic groups or their amides. These include aspartic acid, asparagine, glutamic acid, and glutamine. The next group is with side chains containing basic groups. These include the amino acids arginine, lysine, and histidine. Histidine also contains the aromatic ring, so is also included in the group of amino acids having aromatic rings in the side chain. Besides histidine, this group includes phenylalanine, tyrosine, and tryptophan. The last group is the one having amino acid. This includes the amino acid proline. Next, coming to essential amino acids. Out of these 20 common amino acids, neither human nor any other higher animal can synthesize 10 amino acids in amounts adequate to support infant growth or to maintain health in adults. Therefore, human diet must contain adequate quantities of these nutritionally essential amino acids. These essential amino acids are phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, and lysine. Mnemonic to remember these essential amino acids is PVT, TIM, and HAL. These amino acids at physiologic pH of 7.4 does not remain as NH2 and COOH. Instead, they remain in their ionic form as NH3 plus and COO minus. This condition of a molecule where the molecule contains an equal number of ionizable groups of opposite charge and therefore bear no net charge on it are called Zwitterions. That means amino acids at physiologic pH remain as Zwitterions. And this pH at which an amino acid bears no net charge and thus does not move in a direct current electrical field is called isoelectric pH. Next, coming to the peptide bond. A peptide bond is a chemical bond formed between two molecules when the carboxyl group of one molecule reacts with the amino group of other molecule, releasing a molecule of water. So all the amino acids in a peptide chain are linked together by peptide bond formation, where the carboxyl group of one amino acid undergoes condensation reaction with the amino group of another amino acid, releasing two hydrogens from the amino group and one oxygen from the carboxyl group, giving rise to a molecule of water and forming an amide molecule. This bond between C double bond O and NH is called the peptide bond. Two amino acids which are joined by a peptide bond are called dipeptide. Likewise, three amino acids are called tripeptide and so on. This peptide bond has partial double bond character. Because of this double bond character, there is no freedom of rotation about the peptide bond. And only two bonds are available in a peptide chain, which are shown here by encircling arrows, which have free rotation about the remaining bonds of polypeptide backbone. This imposed semi-rigidity of peptide bond has important consequences for higher orders of protein structure as we will see in further slides. So next comes to orders of protein structure. The first is primary structure, which is nothing but sequence of amino acids in a polypeptide chain. Second level is secondary structure, which involves folding of short contiguous segments of polypeptides, approximately 3 to 30 residues, into geometrically ordered units. Tertiary structure involves the three-dimensional assembly of secondary structural units to form larger functional units. And last is the quaternary structure, which involves the number and types of polypeptide units of oligomeric proteins and their spatial arrangement. Starting with primary structure. The number and order of all amino acid residues in a polypeptide constitute its primary structure. It starts with the amino terminal end to the carboxyl terminal end. The amino acids present in peptides are called amino acyl residues. These amino acyl residues are named by replacing ate or "-ine" suffixes of free amino acids with "-yl". Then peptides are named as derivatives of carboxyl terminal amino acyl residue. For example, a polypeptide sequence having alanine, asparagine, tyrosine, and glutamine is called alanyl, aspartyl, tyrosyl, glutamine. So the amino acid at the carboxyl terminal end is named as it is. The "-ine" ending on glutamine indicates that it is alpha carboxyl group, is not involved in peptide bond formation. The peptide structures are very easy to draw. Peptides are written with residue that bears free alpha amino group at the left, as shown here. First amino group at the left. Then a zigzag line represents the main chain or backbone having the main chain atoms which occur in repeating order: alpha nitrogen, alpha carbon, and carbonyl carbon. Now add a hydrogen atom to each alpha carbon. Next, add a hydrogen atom to each peptide nitrogen. Then add an oxygen to each carbonyl carbon. And finally add appropriate R groups, which are shaded here, to each alpha carbon atom. And the primary structure of peptide chain is complete. Another way is to write the three-letter abbreviations which are linked by straight lines. This represents an unambiguous primary structure. Third way is to write single-letter abbreviations of amino acids in which the lines are omitted. Next, coming to the secondary structure. As we discussed earlier, free rotation is possible about only 2 of the 3 covalent bonds of polypeptide backbone. The angle about alpha carbon and nitrogen bond is termed phi angle, and the angle about carbonyl carbon and alpha carbon is termed psi angle. For amino acids other than glycine, most combinations of phi and psi angles are not allowed because of steric hindrance. So there are only limited combinations of phi and psi angles which are possible in a polypeptide chain. Regions of ordered secondary structure arise when a series of amino acyl residues adopt similar phi and psi angles. So these ordered secondary structures are of two common types, helix and sheet. If we plot the phi and psi angles possible for the polypeptide chain in a graph, we will find that there are two areas where these points cluster. Here the dots represent all the allowable combinations and spaces show the prohibited combinations of phi and psi angles. So the angles of phi between 0 to minus 90 degrees and psi between 0 to minus 90 degree is the area where all the alpha helix structures lies, and the region of graph for phi angle around minus 90 degree and psi angle between 90 to 180 degree is the region where beta sheet structures fall. This shows that the helix and sheets fall within the lower and upper left-hand quadrants of the plot. This plot of phi and psi angles is called Ramachandran plot, after name of the scientist who gave this. So let's come to the two common secondary structures which are allowed by phi and psi angles. First, the alpha helix. In alpha helix, phi angle is approximately around minus 57 degrees and psi angle is of minus 47 degrees. Around 3.6 aminosyl residues are present in one complete turn of alpha helix with a pitch, that means a length of 0.54 nanometer of one complete alpha helix turn. L amino acids, they all form right-handed alpha helix, and only right-handed alpha helix occur in nature. The reason being that only L-alpha amino acids occur in polypeptides. The R groups of each aminosyl residue in alpha helix face outward as shown here. The alpha helix are stabilized by hydrogen bonds between carbonyl carbon of first residue and NH of fourth residue down the polypeptide chain primarily. Besides hydrogen bond, they are also stabilized by van der Waals interactions. Proline cannot form hydrogen bond because of its simple structure. Therefore, it is present in first turn of alpha helix. If it would be present anywhere else, it will result in a bend. Again, because of its small size, glycine also often induces bends in alpha helices. Next, coming to beta sheet secondary structure. Beta means second, recognizable, regular secondary structure. Amino acid residues of beta sheet form a zigzag or pleated pattern, as we can see in this figure also. R groups of adjacent residues point in opposite directions in beta sheet. The beta sheets are stabilized by hydrogen bridges between NH and CO groups of different beta strands. And the peptide backbone of beta sheet is highly extended, as we can see in the diagram also. The interacting beta sheets can be arranged to form two forms of structures. One, parallel beta sheets, where the adjacent segments of polypeptide chain proceed in same direction from amino to carboxyl. Or antiparallel beta sheets, where adjacent segments of polypeptide proceed in opposite directions. Then let's come to turns, bends and loops. Turns and bends refer to short segments of amino acids which join two units of secondary structure, such as two adjacent beta strands of antiparallel beta sheet. A beta turn involves 4 amino-acyl residues as shown in this figure, in which the first residue is hydrogen bonded to the fourth residue, resulting in a tight 180-degree turn. Again, as we know, because of their small structures and simple structures, proline and glycine often are present in the beta turns. Loops are regions which contain residues beyond the minimum number necessary to connect adjacent regions of secondary structures. After this, next come to the third order of structure, that is tertiary structure. The tertiary structure refers to the entire three-dimensional conformation of a polypeptide. It indicates in the three-dimensional space how secondary structural features, which are helices, sheets, bends, turns, and loops, assemble to form domains, and how these domains relate spatially to one another. Folding in tertiary structure is driven by non-specific hydrophobic interactions, and the tertiary structures are stabilized by specific tertiary interactions such as salt bridges, hydrogen bonds, and tight packing of side chains and disulfide bonds. The figure next to this shows tertiary structure of a hemoglobin subunit, where we can see different alpha helices which are connected to one another. The last order of protein structure is the quaternary structure. This involves the three-dimensional structure of a multi-subunit protein and how the subunit fit together. This structure is stabilized by same non-covalent interactions and disulfide bonds as the tertiary structure. Monomeric proteins include single polypeptide chain. Dimeric proteins include two polypeptide chains. Trimeric proteins include three polypeptide chains, and so on. Now, the dimers where two polypeptide chains are present can be homodimers or heterodimers if both the chains are same or different respectively. The figure shows quaternary structure of hemoglobin molecule where 4 subunits are present. This figure shows that 2 subunits are alpha and 2 subunits are beta. So Greek letters alpha, beta and gamma etc. are used to distinguish different subunits of a heterooligomeric protein, and subscripts indicate the number of each subunit type. So for hemoglobin molecule, we will write it as alpha 2 beta 2 heterodimer. Next, let's come to chaperones. Chaperones are nothing but also a group of proteins, but they have functional similarity and participate in folding of over half of mammalian proteins. In the absence of chaperones, protein or polypeptide molecules may undergo damage before they form secondary, tertiary or quaternary structures. But the chaperones, they shield the newly synthesized polypeptides from solvent and other oxidative damages and provide an environment for the secondary structure elements to emerge and coalesce into molten globules, into tertiary structures and into functional quaternary structures. Next, let's come to the classification of proteins. Proteins can be classified into many different categories because of the variety of types and functions and structures of them. But broadly, over here, we have included 3 classifications. Classification based on structure, classification based on composition, and classification based on function of proteins. Let's first come to classification based on structure. Based on structure, proteins can be of three types: fibrous proteins, that means which are in the form of fibers; globular proteins, which are in the form of globules; and intermediate proteins, which include all other structures of proteins except fibrous and globular. The globular proteins, as you can see in the figures shown, are folded into a compact spherical or globular shape and are soluble. Fibrous proteins are long strands of fibers, elongated, tough, and insoluble. The globular proteins form several types of secondary structures, and fibrous proteins form single type of secondary structure, that is, sheets. Globular proteins have various cellular functions. They act as enzymes and globular proteins, while fibrous proteins are primarily involved in structures formation. They are used in support, shape, and external protection. Over here is shown an example of carbonic anhydrase enzyme, which is a globular protein, and collagen, which is a fibrous protein used in structure formation. Second classification is based on composition. Based on this, proteins can be simple or conjugated. Simple proteins are devoid of structural constituents other than amino acids. For example, albumins, globulins, protamines, etc. Conjugated proteins have other non-amino acid components as well. Example, glycoproteins have carbohydrates and amino acids. Lipoproteins have lipids and amino acids. Nucleoproteins have nucleic acid components along with amino acids. Metalloproteins and flavoproteins have metals along with amino acids. And phosphoproteins are phosphorus plus proteins. Third classification is based on function. And based on function, proteins can broadly be categorized as structural proteins, enzymes, hormones. Second, as pigments, transport proteins, and contractile proteins. And thirdly, as storage proteins and toxins. And all these are self-explanatory in itself. Lastly, let's come to the biological importance of proteins. I'm sure we all know a lot about the biological importance of proteins. Let's just compile it all in one slide. Proteins function as signaling molecules. The cytokines in our body, they are all proteins which communicate with other cells to give signals. Proteins form major parts of blood. Hemoglobin is a protein molecule, as we saw. Its role is to transport oxygen. As fibrinogen, proteins perform the role of blood clotting. As albumin, protein maintain proper amount of liquid in blood. Proteins also form enzymes. The digestive enzymes, other hormones in the body, they are all proteins which help in breakdown of food, especially when we talk about digestive enzymes. Proteins form major component of cell membrane. They form channels for substances to move through the membrane. They act as enzymes. They act as receptors. The three different types of proteins in cell membrane, the peripheral proteins, the integral proteins, the lipid-bound proteins, they all help in transfer of substances in and outside the cell. Proteins form the major structural parts of our body. The cytoskeleton, that is the skeletal component of each cell, then are nothing but network of protein filaments and tubules which maintain the shape of cell. Keratin, found in skin, hair and nails, are nothing but proteins. Collagen provides strength, that is also protein. Elastin, which provides flexibility to our body, is also nothing but protein. Proteins in muscles, we all know when people go for a gym, they take protein shakes and protein rich diets because they want to enhance their muscles. So proteins form major part of muscles. The actin and myosin filaments of muscles are nothing but proteins which interact with each other to enable muscle movement. Myoglobin are also proteins which release oxygen to muscles. Ferritin, which store and release oxygen, are also proteins. Proteins in the immune system, the antibodies which fight invaders and infection, are nothing but protein molecules. The complement system, having 20 protein molecules which are activated during infections. So all in all, we can clearly see how protein are so important for us, and we must always take protein rich diet. Overall, I would say we would thank protein for building and repairing this awesome body of ours. We would not have muscle, bone, hair, nails, or lips without proteins. That's all for this lecture. Thank you so much.